Hello. I'd like to thank the organisers for the opportunity to tell you about the non-aversive handling methods that we've developed for mice, a refinement that has a big impact on mouse welfare, as well as reducing the confounds of unnecessary stress in animal research. I'm going to talk through some of the scientific evidence that shows what a big effect these handling methods have on mice, and then look at the practicalities of using these methods in place of tail handling and some tips on how to implement them successfully. But first, domesticated mice are a lot easier to handle, of course, but we do know that background stresses in the laboratory environment can have a major impact and confound the repeatability of research. When we started to use laboratory mice as genetically controlled models for wild mice in our own research, I was really surprised by how anxious they were and how much that interfered with our behavioural experiments. And indeed, when we were running experiments, we found that we needed many more individuals of an inbred strain than we did of wild mice to show the same level of effect. But why should domesticated laboratory mice show such high anxiety when all we were doing was actually handling them and putting them into simple behavioural tests that shouldn't have been aversive? I'll just give you a little. So, wild mice do get very stressed if you pick them up by the tail in the standard way that laboratory mice are handled. So to see if this tail handling is what was causing the problem, I compared 3 different methods for picking up mice, and the differences were really quite astounding. So that led to us conducting a formal study, which was published in Nature Methods in 2010, because it turns out that the method that you use to pick up mice is critical to whether this causes handling stress and anxiety. And it's this paper that won the NC3R's prize in 2010, and the work that we've done since then to show that these methods can be practically applied in laboratories won the Swiss Laboratory Animals Association Refinement Prize in 2019. So what are these methods that we're comparing? A little background. So the first method is the traditional tail handling method, where we pick the mouse up by the base of the tail between thumb and forefinger. And we're not dangling these mice, we're using the best practice of supporting the weight of the mouse on the arm or the sleeve or the hand once the animal has been picked up. And we're comparing that to a tunnel handling method, which is based on the method that we use for wild mice. So here we're encouraging the animals to enter a tunnel and picking them up in the tunnel. And then you can either use that to transfer the animals to another location, or if you need them in the hand to conduct procedures or to do health checks, you can just tip them backwards out of the tunnel onto your hand and then replace them back in the cage. And then the third method that we've used is cupping them on the open hand. Domesticated mice, of course, don't show the same flight and jumping that wild mice do, and you can just pick them up on the open hand without restraint once they're used to being handled. So once they're used to being picked up, they will stay on the hand when you lift the hand. Getting them to stay on the hand in the first place does require a few little techniques to do that, and I'll say a little bit more about that later in the talk. So we've compared those 3 different handling methods, and over the past 10 years, we and other labs have assessed the effects on mice from many different angles. We've looked at many behavioural tests like voluntary interaction, how easy they are to handle, the anxiety they show, also physiological stress responses, how they respond to rewards, the reliability of both behavioural and drug testing, and also how the animals respond to different procedures. Very unusually, all of these studies have come to the same consistent conclusion, which is picking up mice by the tail has negative effects on animals that can be avoided by using our alternative methods. Now, I won't have time to go through all of this evidence, of course, so I'll pick out some of the key findings. And one thing that I'm particularly interested in is whether animals find handling aversive or whether they will voluntarily interact with the handler. So it's telling us something about the way the animal itself feels towards that handling procedure. So to look at this, we do a very simple test. Immediately after handling, we put the handling device back into the cage and hold it unmoving so we can see whether the animals will approach and interact voluntarily. And these mice have just been picked up by the tail, and you can see that they're really unwilling to come and interact with the hand. This mouse does seem to be trying to escape from the cage to avoid the hand. And when they do actually approach the hand, it's a really cautious type of approach. They sort of sniff forward and then they back off. Very different from the way that these animals on the right-hand side now have just been handled in a tunnel, and the first thing they do when you put the tunnel back into the cage is go straight back into the tunnel. They're much more willing to approach the handling device, and as you can see here, they'll touch it, they'll actually climb on it, so they'll put all their weight on it, and they'll go straight back inside the tunnel. So a much more free interaction as they're exploring the cage. They're not just sitting in a corner. Started to look at this graph on the right just summarizes the total time that mice spend interacting with their handling device across 3 different strains and 9 different handlers. And you can see there is a substantial difference between the different methods. Mice that were picked up in the tunnel, the blue bars there, spend a lot of the test time interacting with the handling device, the tunnel, and the hand holding the device. Whereas the mice that were picked up by tail spend very little time interacting with the handler. Mice picked up by cupping show a sort of intermediate response. They're actually showing a lot more time interacting than those picked up by the tail, but not quite as much as those picked up by the tunnel. And particularly the male mice, they show a little bit more caution about interacting with the hand after cupping compared to the females. This difference in their willingness to interact with the handler generalizes across strains, across handlers, whether handlers are really experienced or inexperienced, whether they're male or female. It generalizes across cage types, across different laboratories, and even whether mice are handled in the light or the dark. We really see tremendous reproducibility in these responses across many different studies. Really unusual to see that consistency in a behavioural response across so many different factors. But in this test, the tunnel-handled mice, of course, are responding to the tunnel, while the tail and cup-handled mice are responding to the hand. But for the tunnel-handled mice, it's not just the tunnel that they're more willing to interact with. If we test the tunnel-handled mice and look at their response, even if we just put the hand back in, we see that actually they're also a lot more willing to interact with the hand than the tail-handled mice are. These animals are essentially just much more willing to explore, and they will start to get used to the hand, and they'll start climbing on the hand and even climb up your arm if you'll allow them to. Whereas the tail-handled animals are showing a much more aversive response. They spend very little time interacting with the hand, and they're very loath to actually touch the hand. So we see a really different response between these different methods. So as well as interaction, we can see big differences in response in lots of other measures that we can. Zoologists. So one of the measures that we've looked at is urination and defecation during handling, because animals which are highly stressed show much increased rates of urination and defecation, and it's something we can compare easily between the different methods. And what we see is that the mice that are picked up by the tail show much increased rates of urination and defecation, a really typical sign of quite a high stress response. It's not just us that have shown this. A number of other studies that have also repeated this and measured urination and defecation and find exactly the same thing. Now, I was particularly interested in looking at anxiety because it was this that really alerted me to the potential problem of picking up mice by the tail. So what we wanted to test is whether that actual picking mice up by the tail causes that high anxiety response that we notice in laboratory mice. So we've used a number of tests. Perhaps the simplest test is an open field test. Anxious animals will avoid going into open areas. And in an open field test, we deliver the animals into the center of an open field and then record their behavior over a 5-minute test. And what we see is that tail-handled animals spend much less time in the center of the arena than those handled by tunnel or cupping. And they're also much slower to return to the center once they've been released into the arena. And what I want you to really notice here is that these aren't small, subtle differences. They're really big. This is a very major difference in behavior between these animals. Certainly between the tail and the tunnel and cupping methods, we find no difference actually in anxiety behavior between those that have been picked up by tunnel and those that have been picked up by cupping. We've also looked at behavior in an elevated plus maze as a really well validated test of anxiety. And again, the tail-handled mice consistently show greater anxiety in this test. So the elevated plus maze has 2 closed arms, which are surrounded by high walls, and 2 open arms. And the tail-handled animals spend much less time on the open arms than those handled by tunnel or by cupping. And they show a lot more risk assessment behavior, where they sort of sit in the center of the maze and just stretch out onto the open arms without actually going there. A very typical sign of anxious animals. Interest in one. And indeed, these tests have been repeated by many other people, and there have also been additional tests, social novelty tests and light dark box tests. And all of these studies have found the same thing, that mice picked up by the tail have higher anxiety compared to those picked up in a tunnel or by cupping on the open hand. So even though typically we see quite a lot of variation in these types of behaviours between strains, between sexes, between laboratories, on this particular factor, how the animals are handled, it's really consistent. Rodent behaviour. Now, besides the obvious impact of these differences in anxiety and stress on animal welfare, uncontrolled background stress can cause major confounds in scientific experiments and reduce data reliability. So we tested the mice in a behavioural test to see whether the method of handling actually influenced the reliability of the behavioural outcomes of the test. And we decided to use a very simple test that any mouse should be able to do. This is an dishabituation test, which tests the ability of mice to recognise a difference between 2 stimuli. And in this test, what you do is you present the mice with the same stimulus in a series of tests conducted quite quickly after each other. And as the animals start to recognise the same stimulus, their investigation declines. And then on the 4th trial, you replace that stimulus with a novel stimulus, and if the mice recognise that that stimulus is novel, then their investigation of it increases, and it's this dishabituation, or so-called dishabituation, that the test is measuring. So it's the difference between their sniffing of a very familiar stimulus with a novel stimulus. And to make this really easy for the mice, we used urine cues. Mice are extremely good at discriminating between urine cues. So we used urine cues from genetically distinct individuals, so they should really be able to tell that difference very easily and communicate. So here you can see the results for mice that were handled by tunnel. Here you can see that they show a dishabituation, an habituation as they repeatedly get presented with the same stimulus. And here's the response we're looking for when we swap the first urine stimulus for another urine stimulus from a different individual. Then they recognise it's different, and we see the dishabituation response. Really nice and consistent. Every single mouse in the test could do it. These are the responses for mice that were handled by cupping. And here we can see that, yes, we do see a dishabituation response. It is significant, but you can also see that we've got much more variance in these animals. It's just significant, but also some of the animals didn't actually respond properly to the test. So we did see a response a little bit more variable. And that's quite typical actually for cupping, because in these tests, we didn't subject the animals to a lot of handling. Their only prior experience of handling was during cage cleaning every fortnight, and then when they were put into the test. And we know that for cup handling, animals do take quite a lot of experience with cup handling before they start to show very easy responses to it. So that's pretty much what we might expect to see for cup handling. Now, these are the results for mice that are handled by tail. And I haven't forgotten to plot the data here. I've plotted the median responses. And the median responses of these mice was 0 investigation. The test failed because the animals failed to investigate the stimulus. And this is really typical of this test. When people try doing this test on laboratory mice, it's a test that is very difficult to carry out in a novel test arena where you have to handle the animals to put them into the arena. And indeed, people have completely failed to get this test to work in novel arenas and have ended up having to singly house the animals so that they can conduct the test in their home cages without actually handling the animals. But actually, if you handle the animals using a non-aversive method, they will do this response in an unfamiliar test arena really, really well. The actual problem here is that the animals are responding with anxiety in the test. And when we look at the behaviour that these tail-handled animals were showing, it is typical of the behaviour of a really anxious animal. They're staying around the outer walls, they're not exploring the enclosure properly, and they're just failing to investigate the stimuli. And indeed, even when we put the stimuli against the side walls where the animals were spending most of their time, they still failed to investigate the stimuli. The anxiety was just really interfering with them showing a response. Now, my laboratory, of course, is interested in behaviour, and many others are more interested in looking at the effects of physiological stress and whether stress from handling can interfere with their physiological experiments. So this is data from a laboratory that studies diabetes, where stress is a major confounding factor that affects blood glucose and glucose tolerance tests. So this laboratory compared tail handling mice with cupping them on the open hand and found that the cup handling significantly reduced both the blood glucose and corticosterone stress responses of the mice. And on top of that, it also improved the glucose tolerance responses of the animals. So it really had quite a big effect on these animals, showing that cup handling was a much more reliable technique to use in these studies. On top of that, Jasmine Clarkson has looked at the effect of handling on chronic stress by measuring the adrenal gland weights after mice have been handled, either by picking up by the tail or in a tunnel, and showed that the adrenal glands are much heavier in tail-handled mice than tunnel-handled mice after a few weeks of handling, suggesting there really is a chronic stress response to tail handling. Now, one of the things that worries people about using something like tunnel handling is that the mice don't get used to being restrained. And the same with cup handling. Although you're putting them on the hand, they're not actually being physically restrained. But of course, for many procedures in the laboratory, we do need to restrain animals. So I think that the concern is that once animals start to have to be restrained, we'll then get a very strong response to that restraint. But that actually isn't the case. What you can see here is an experiment where we have handled the animals by their standard method, tail, tunnel or cup, and then immediately after they've been handled, we've then restrained them by the scruff in a standard way, and then we've measured their voluntary interaction with the handler again immediately after that scruffing experience. And actually, the only animals to respond in a negative way to scruffing are the tail-handled animals. So these animals were already showing a poor response. Once we've restrained them by the scruff, they're showing an even more negative response in not being willing to approach the handler. And that is really very typical. It's the tail-handled animals, or even though they've had experience of restraint, when you give them another experience, actually they respond really strongly to it. Whereas this really didn't affect the tunnel-handled animals or the cup-handled animals, and they showed no more anxiety than they did before we did the scruffing. So they're very tolerant, actually, of being restrained by the scruff because we've got them used to being handled in a non-aversive way to start with. And importantly, actually you can restrain the mice by the tail once you've picked them up. That itself isn't aversive. It's only being picked up by the tail that induces this aversive response. So once you have the animal in the hand, whether you pick them up in a tunnel or by cupping, you can then restrain the animals by the tail or by the scruff. It's particularly useful to be able to restrain the animal by the tail if you want to inspect its abdomen, for example. That isn't going to cause you a problem. It's the way that you're picking animals up that causes the issue. I often study animals. So there is a lot of evidence now that picking up mice by the tail is highly aversive and causes substantial stress and anxiety. These non-aversive methods, on the other hand, really seem to overcome that. But is it really practicable to use these non-aversive tunnel or cupping methods for routine handling in the laboratory? I think one of the things that is particularly of concern is how long do we need to handle the animals to get them familiar with these non-aversive methods before they're effective? And do we need to have tunnels in the cage? Certainly in all our experiments, we provide animals with tunnels in their home cage, but is it actually necessary? Could you just use a clean tunnel to pick them up? And I think one of the things that is particularly of potential concern is even if we use non-aversive methods for routine handling, once animals are used in procedures that might induce some pain or distress, is that going to completely negate the effect of the non-aversive methods? So Kelly Gouveia came to work with me as a PhD student to try and address some of these practical issues. In semi-natural. I think some of the thing of particular concern, particularly if you are an animal care person and you've got to handle the animals on a regular basis, that mustn't take too much time. And when we first started doing this work, we followed the standard practice of assuming that if we wanted to familiarise animals with handling, we needed to pick them up and hold them for a reasonable period of time so they could get used to it. But the good news actually is that you don't need to do that. If you use a non-aversive method for picking them up in the first place, that in itself is good enough. You don't have to then hold the animals to get them used to being handled. You just have to not use an aversive method. So what we found that there was no difference in whether after we'd picked them up, we held the animals for just essentially 2 seconds, about the time it takes you to transfer an animal from one cage to another, or whether you use much more extended periods. So the time of handling actually doesn't seem to have any real effect. And also how frequently you handle is not that important, at least for tail handling and tunnel handling. So if you tunnel handle at cage cleaning, that is sufficient to get a positive response, even if you're only cleaning the animals out only quite infrequently, maybe on a 2 or 3 weekly basis. And what we found was that, on the other hand, the bad news is that tail handling induces a negative response, even if you handle them only really briefly and perhaps only at cage cleaning. That will still induce this really strong negative response. But for tunnel handling, if you can just pick them up in the tunnel and they start to respond really positively very quickly. Cupping them, on the other hand, does take a little longer. Here you can see some data where we pick the animals up for different periods of time, and the difference of periods of time don't really make very much difference. Certainly the 2 seconds had the least positive response, but really we weren't getting a very good response after 5 handling sessions. And this is very typical of cup handling, especially with the slightly more nervous strains. What we found is that you need to have more than 5 handling sessions for cup handling for animals to become really accustomed with cup handling. Typically, it takes them about 7 to 10 handling sessions before they get comfortable with this method. So that's one of the slight downsides of cup handling. It's very quick to do. You don't need to have a tunnel to do it, but it takes the mice quite a bit longer to get used to that compared to tunnel handling. Natural enclosure. Now, tunnels, as I said, when we do, in our cages, we have tunnels for handling present in the home cages, so that animals can get very familiar with these tunnels before we ever have to handle them. The question is, do you need to have a tunnel in the home cage? In some cases, it may be quite difficult to have that tunnel, especially if you have quite small cages and there are other things that you want to have in that cage. What we found is that home cage tunnels do provide additional enrichment in the cages, so the animals use them quite a lot. And one of the advantages to that is that the animals get so used to them, it's very easy to get them to enter the tunnels very readily. But in terms of the actual responses of the animals, if you can't have a home cage tunnel, you still get very positive responses to unfamiliar external tunnels that you would just use for handling them. It's still a lot better than tail handling, but there are some additional advantages if you can have the tunnel in the home cage, especially for the more nervous strains. But the mice will get used to going into a clean tunnel really very quickly. So it's perhaps particularly beneficial if you're not handling them very much. A home cage tunnel will just help that little bit more. To model. So what we've seen is that these methods are actually quite practical to use. You don't need extended handling to make use of them. But is any of that of any use at all if the animals then go into an experiment where they're experiencing procedures that may induce some distress or pain? So one of the things that we've looked at is the response of animals once they have experienced a particular procedure. And here you can see an experiment where we've looked at the response to subcutaneous injection. So here we've got 4 different groups of animals. It's a little bit more complicated, so I'll just show you. At the top, you can see the results for voluntary interaction. At the bottom, we have an open field test where we're measuring anxiety. For the 4 different groups, the hatched bars show animals which have been picked up from their cage, restrained by scruff, and given a subcutaneous injection. And this is their response after their first injection, and this is after their 5th daily injections. And the control animals here in the solid bars have just been picked up from their cage by their normal method and then replaced. So they've not been restrained, they've not had injections. And here you can see we have tail-handled animals in those 2 categories and tunnel-handled animals. And what you should be able to see very easily is there is a massive difference in response, again, according to the method by which they've been picked up. The tunnel-handled animals are showing much more voluntary interaction than the tail-handled animals, and they're showing much less anxiety than the tail-handled animals after both first injection and 5 injections. And actually, we can see no response to the injection. Even after 5 daily injections, we still couldn't see any response to the actual injection itself. The handling method was having all of the effects here. So although we typically would think of injection as being something that the animals are going to respond quite badly to, it turns out that the handling method is far more important. Lindsay Henderson has also recently published a study where they show very similar effects, that tunnel handling reduces negative responses to IP injection or to anaesthesia compared to those picked up by the tail. So this does seem to be quite a general type of response. Natural condition. Here you can see something which perhaps involves the animals in a little bit more obvious distress. This is the response to daily gavage, which is used for drug administration. This is a study by Nakamura and Suzuki, where they compared the responses of tunnel-handled animals with tail-handled animals when they had to give them a daily gavage. And what you can see here is that clearly once they started doing daily gavage, the voluntary interaction of their animals did drop, but it dropped in both the tunnel and the tail-handled animals, and still the tunnel-handled animals were still coming to the handler and interacting voluntarily. Although they were a little bit more wary, it was still a much stronger response than the tail-handled animals, which really started to avoid approaching the handler. So you see this much better voluntary interaction. They also found that the tunnel-handled animals were easier to handle using an independent rating scale. They showed that even despite the experience of daily gavage, the animals, tunnel-handled animals, still showed reduced anxiety. And perhaps particularly importantly, they found that the tunnel-handled animals had reduced variation in pharmacological testing. So this was a real benefit from the study, not just for the welfare of the animals, but also improved their pharmacological tests and behaviour. So the good news from all of that is that procedures don't reverse the positive effects of using non-aversive methods, even if those procedures do involve a degree of distress or potential pain. And they really are very practical to use. You don't need to spend a lot of time handling the animals. You can just replace tail handling with these methods. But I think for people who have always picked up mice by the tail, there are a lot of worries about changing to these non-aversive methods. I think in particular, people do worry that it's going to take them more time, that it's going to be more awkward, they won't be able to do some things, maybe they won't be able to handle as easily if they've got small cages. Lots and lots of worries. I think inevitably thinking about a change does immediately make you think of all the potential problems. And I think there is an immediate assumption when you see these techniques, they're just not going to be as practical as picking up mice by the tail. So to help, we have partnered with the NC3Rs in the UK so that we can provide people with a lot of resources, and we have set up a special mouse handling hub on the NC3Rs website. I've given you the URL here. And here we put a lot of resources, like we have made a mouse handling tutorial that will take new handlers all the way through the use of mice and some of the initial scientific evidence, as well as tips for how to handle the animals. But there's also a lot of other information there, not just the tutorial, but short video clips that you can use in training. And I'm going to show you a few of those just to show you some good tips to doing this handling well. You'll find frequently answered questions, looking at how you can practically implement these in laboratories. There's a summary of all the research papers, not just the bits I've shown you here, but actually it summarises all of the research that we know that has been done on these methods. And you can also get a poster free from NC3Rs if you contact them. It's a poster to put up in your animal facilities, giving lots of hints as to how to best implement these methods, not just what to do, but also some of the things perhaps to try and avoid for good practice. I think the real key here is making sure that people have good skills in learning these new handling techniques, that they can actually handle as well with the tunnel as they can with the tail before you roll it out. What you don't want to do is sort of start doing this and start thinking that you're going to be able to do this straight away on a lot of mice, and it's not going to take you more time. It will. I'll be quite honest, it usually takes it could be a few days, it could be a few weeks, depending on your skills, to learn to do these techniques really, really well. But once you have learnt these techniques, you should be able to do them as quickly as picking up by the tail. You also need Now, I know that people are often sceptical to start with, that they're really going to see a big difference, and this really is a practical thing to do. My own chief animal technician, John Waters, was exactly the same. When I first started this research and we found the results, I asked him if we could implement the new methods in our own animal facility, and he really didn't think it would be practical. He just thought this was going to ruin his daily life. He would never be able to fit in the work. But he agreed to give it a go because he'd seen the data and he could see what a big effect it was having on the animals. And since then, he has been a massive champion. I mean, John really worked out how to do a lot of normal things around the unit by using these non-aversive techniques, and he's helped a lot of other technicians to be able to implement things. And because of that, he has very well deserved a number of awards himself, and really seen this from a sort of technician's perspective as to how to do this on a daily basis. And we hear very similar things from those people who have changed. In particular, people say it's not as difficult as they anticipated. They particularly recommend everybody should train first and implement slowly, so that you are really good at the handling techniques before you try and take on a lot of animals. I think people worry about whether they can do the health checks in the same way, but health checks are fine once you adapt to the new way of handling. Always people report animals are more relaxed, they're easier to handle, and they get fewer bites. And in particular, new trainees are much less scared of handling with a tunnel. So trainers tell us that it's much easier to train new trainees because they're not scared of the animals, and not just in using these techniques, but also because they're not scared of the animals. They find doing things like scruffing and restraint of that sort much easier to do. And I think one of the things perhaps I wasn't expecting when I was doing this, because I was really just thinking of the animals and also the impact on the science, is that people say it really increases their personal satisfaction. John has certainly said that this is a really big thing for him. He feels much closer to the animals. The interaction with the animals is so much better that people just say they would not go back to tail handling. Use control. And indeed, if you want to hear what people think that have implemented these new techniques in their own facilities, NC3Rs has published some interviews from technicians and training officers who have implemented these methods in their own facilities. And they talk a little bit about their experience of how to do this in a way that people find easy to take on. So if you have chance, I would definitely recommend you listen to some of those people. I think they're really quite inspiring. It's certainly something to take on, particularly if you have colleagues who are just not so sure. They haven't listened to a talk about this. It just sounds like more work to them. Then perhaps hearing other people and hearing that they found this a really positive experience might be a useful thing for them to hear. Laboratory conditions. So finally, I think perhaps the most important thing I can tell you is how to implement these methods successfully. Is you really do need to get good technique. I think it's one of those things that while you see the videos, it sort of looks interesting, and then you get a tunnel in your hand and it doesn't go quite so well. So I'd just like to sort of show you some good tips for how you actually handle with a tunnel and how you get the animal quickly into the tunnel without any fuss. So I'm showing you in a big cage because it's actually slightly harder to handle in a big cage. And you can see there, I've held the tunnel against the side wall of the cage. And then you use the other hand to encourage the animal into the tunnel. Now, what you don't want to do is to put that tunnel in the centre of the cage, because now when you encourage the animal to go in, it's just going to run straight past. I don't think this mouse knows it's supposed to go into that tunnel. It's just seen the hand coming towards it and gone in the opposite direction. So you need to just if you put the tunnel against the side wall, there's nowhere else for the mouse to go. And notice we're taking the mouse to the tunnel, not the other way around. Now here you can see that you're just using the hand to block. You're not actually using the hand to encourage the animal in. That's not going to work. You can see this mouse is just getting frustrated. It's been blocked into a corner and doesn't particularly want to go in the tunnel. What you need to do is just encourage the animal into the tunnel, and indeed if it doesn't go straight in, and many animals will, but if it doesn't, then you just touch the back of the animals and it will go straight in. Now you don't want to do this. Do not chase the animal around with the tunnel. It never works, or only very rarely works. It's a really you'll get very, very frustrated if you do that. Instead of that, you are holding the tunnel still and bringing the mouse to the tunnel. It's very, very tempting to, when the mouse moves, to move the tunnel with it, but try not to do that. Hold the tunnel steady, get used to taking the mouse to the tunnel, and you will see that works really quickly every time. To drill down. So here is an example of cleaning out a cage. It's an IVC cage using the tunnel handling technique. You can see there is already a clean cage set up by the side with a clean tunnel in it, and we're cleaning out the cage on the left-hand side. Sorry, on the right-hand side. And here you see the tunnel goes in. other hand is used just to encourage the mouse into the tunnel. Now, because we're using clear tunnels, it's very easy to actually inspect the animals in the tunnels. And one of the nice things about using a clear tunnel is that the animal is free to move around. So you can really see if there is anything wrong with that animal. If you want to inspect it more closely, of course, you can tip it out onto your hand. But here we're just doing some inspections and just checking that the animal is fine. And you'll notice that when they go into the new cage, they go straight back into the clean tunnel that is in there. They have no fear of these tunnels at all. Very easy to get them in. Now, I've paused the video here because actually I want to see what happens when you don't place the tunnel correctly and actually make it such that the mouse can go straight past you. So what you're going to see now is that the handler puts that tunnel in a different place in the cage, but they've not prepared it correctly so that they're not blocking the animal from going elsewhere. And just see what happens. Into the mechanism. So here you see now the tunnel's gone over onto the right hand side, but there was nothing blocking the animal going in the other direction. And now we're getting a little bit of chasing the animal around, and only got a good handling once the tunnel was back into a secure place and the animal was brought back to it. Let's just perhaps see that again so you can see what happened. See, the tunnel went into a place, the animal just avoided the tunnel entrance, it wasn't properly blocked to go, so it couldn't go past. And now once we get the animal into a secure place for handling, we can very easily get it in. And I think I would recommend to you when you first start that you practice, it doesn't matter which hand you hold the tunnel in, whichever is most convenient for you, so that you can use the other hand, but practice so that you can very easily isolate that mouse into one corner and push it into the tunnel. Once you get good at that, then try with other placements of the tunnel around the cage so that you can work out the best placement of the tunnel and your hand. And you will gradually find that as you get better and better at this, you will soon be able to place the tunnel correctly in different parts of the cage to very quickly get the mouse wherever it is. But I think when you first start, it's a lot easier to get the mouse to come to the tunnel in a standardized place and get really good at that first. Involved. Now, what about cup handling? Now, on the surface, cup handling looks really easy. You're just going to scoop up the animal onto your hand and hold it there as you lift it up. The problem with cup handling is it is a little harder because you need to understand how to get mice to stay on the hand and how to recognise if a mouse is not going to stay on the hand, because we don't want mice jumping all over the place. So cup handling is suitable for perhaps older animals, animals which are already familiarised with handling, and not so suitable for young jumpy animals, especially those animals just first after weaning. And I would say that cup handling isn't really good for new handlers. I think they need to start to understand mice a little bit better and get used to handling them before they use this technique. But for more experienced handlers, this is a very convenient technique. You don't necessarily need a tunnel to do it. But the trick is, how do you get mice to stay on the hand before they're familiar with this technique? And there are a number of methods that you can do. And I think this method is probably the easiest. It is using a tunnel to familiarise animals to being picked up in the first place. So here I will show you. These animals have never been cup handled. And what we do, first of all, is get them used to being picked up in a tunnel. And when they're steady in a tunnel, they stop sort of showing any anxiety about being picked up. Then you can tip the animals out backwards onto the hand and they will stay there. But don't do that before the animals have got used to being picked up. But the animals that get picked up in a tunnel get very relaxed. They're not anxious animals. And so once they've got used to the tunnel, and I would say pick them up in a tunnel maybe 3 or 4 times before you attempt this, and then you can tip the animals backwards onto your hand. Now, that's fine for older animals. If you've got really jumpy animals, they will still try and jump off the hand. So you do need to be able to recognise that in your animals, and don't try cup handling for those animals which are still rather jumpy. But for this is essentially an alternative to tunnel handling, and in our lab we use the two interchangeably. So we get animals used to being picked up in a tunnel, and then we can either pick them up in a tunnel or scoop them onto a hand. If we're using them in a behavioural test, we will always pick them up in a tunnel because it is just that much less likely to cause any anxiety in the animals, and we've then got really calm animals going into our tests. But for routine handling, if you want to inspect the animals and it's quicker for you to pick them up on the hand, then get them used to being picked up, and then you can just scoop them up onto the hand. Now, there is another technique, and that is to, the first few times that you pick up the animals, instead of picking them up on the open hands, just pick them up between closed hands, very loosely held around the mouse, and you will feel the animal sort of trying to push between your fingers and trying to get out. So wait for the animal to calm down. It should take about 5 to 10 seconds, no more than that. And if it then calms down, you can open your hands and just check on the behaviour of that animal. And as long as it's not a really jumpy animal, it will stay on the hands. So you can do this and just be you will need to watch the animal, but if you're an experienced handler, you should find this works pretty well. And I know people get very concerned that they might get bitten when they do this, but really if you're approaching the animals very carefully and you're not squeezing the animal, it'll just try and push between your fingers. But I have to say I have not been bitten like this, except when an adult male weed all over my hand, and then I forgot to clean my glove before I handled an unfamiliar male, and he bit me. I think it was a response to the urine, rather than being cupped on the hand, because no other mouse has ever bitten me picking them up this way. And another method you can use is when you're transferring mice at cage cleaning, you can just scoop them and transfer them between closed hands in this way. And once you've done that for several times, the animals really start to calm down, and you will find that you can just scoop them on open hands after that. But I would just warn you, please don't try this technique with jumpy animals, particularly with young animals, or if you've got any particularly jumpy strains that are always difficult to handle. Tunnel handling is a lot more secure, and I would really recommend that. So some take-home points from my talk. Picking up mice by the tail induces strong handling aversion, stress and anxiety. This negatively impacts animal welfare, and it's a really surprisingly strong response. The animals are harder to handle, and it can be a major confound in research which is sensitive to stress, and an awful lot of research we know is indeed sensitive to stress. However, that can be easily avoided by picking up mice with non-aversive handling methods. And we've shown that this is really highly repeatable. It really does seem to work on lots of different strains, both sexes. It doesn't matter whether the handler is male or female, whether they've had a lot of experience or very little experience. We always seem to get the same responses in using these non-aversive handling methods. So I particularly recommend that you use a tunnel, or perhaps other objects within the home cage may work also. You can cut them on the open hand once the animals are habituated to being picked up. But do bear in mind that the first thing the animals need to get used to is just being picked up from the cage. And until they get used to that, they are quite jumpy. Fortunately, you can still restrain them by the scruff or by the tail once you've picked them up using a non-aversive method, and you can still conduct procedures even if those do induce some pain or distress when you need to. It's not going to reverse the response to the non-aversive handling methods. And I think we have shown that these are easily incorporated into normal husbandry. It should take you little or no longer than tail handling once you are competent, but it does take you a little time to become competent in the first place. So you will need to spend a little bit of time just getting your techniques really good and making sure that you can handle really quickly and easily using these methods. The mice themselves only need brief familiarisation, particularly with tunnels. We don't need to handle them for prolonged periods to get them familiar with handling, as long as we use a non-aversive method to pick them up. Cupping on the open hand does require more handling experience, and the response of the mice is strain and age dependent. So use that with caution, use it when it's safe, otherwise use a tunnel, because that seems to work in all situations. And there are real strong benefits to using these non-aversive techniques. It's better for animal welfare. The animals are easier to handle. We see improved reliability of response measures, particularly where these responses are susceptible to stress effects. And also for the handlers, we actually see improved job satisfaction, because people really enjoy that much better interaction with their animals. Instead of the animals trying to run away from you, they come to you. They actually want to climb on you. They want to interact. And that has to be good for everybody. natural. So I'd just like to finish by acknowledging the very many people who have helped with this research, particularly all the people who have helped with handling the animals in my own group and have been very good about handling the animals and letting me record the responses of the animals. Also particularly to NC3Rs for helping particularly to put together resources to train people and that can be used by others. To other collaborators, particularly to our funders, and of course to the mice themselves, without whom this research would not have been possible. And thank you for listening. Thank you. Behaviour in wild rodents under laboratory conditions is challenging, but wild mice will actually cooperate surprisingly well with the careful approach and handling methods that we've developed.